Welcome to this lesson on plant hormones. Plant hormones are fascinating chemical compounds that act as growth regulators in plants. These molecules control nearly every aspect of plant development, from seed germination to fruit ripening. Throughout this video, we'll explore the major types of plant hormones and understand how they coordinate the complex processes that allow plants to grow, respond to their environment, and complete their life cycles. Plant hormones are chemical messengers that are produced in very small quantities within plant tissues. Despite their low concentrations, they have powerful effects throughout the entire plant body. These hormones are synthesized in one location, such as the shoot tips or roots, and then transported to target tissues where they trigger specific responses. One of their most important roles is regulating how plants respond to environmental stimuli like light, gravity, and stress conditions. Think of them as the plant's internal communication system, coordinating growth and development across all its parts. Let's begin with auxins, one of the most important classes of plant hormones. The primary function of auxins is to promote cell elongation, which is essential for plant growth. They also control apical dominance, which is the phenomenon where the main central stem grows more strongly than the side branches. Auxins are responsible for tropisms, the directional growth responses like phototropism, where plants bend toward light, and gravitropism, where roots grow downward. The most common naturally occurring auxin is IA, or indole-3 acetic acid. This hormone is produced mainly in shoot tips and young developing leaves, and it moves downward through the plant to reach other tissues. Gibberellins are another crucial group of plant hormones. Their primary role is promoting stem elongation and overall plant growth. Gibberellins are particularly important for seed germination, as they trigger the breakdown of stored nutrients that the young seedling needs to grow. They also help break dormancy in buds, allowing them to resume growth after periods of rest. In addition to these functions, gibberellins can induce flowering in certain plant species and promote fruit development even without fertilization, a process called parthenocarpy. Interestingly, gibberellins can overcome genetic dwarfism in plants, causing dwarf varieties to grow to normal heights when treated with this hormone. Cytokinins get their name from their role in promoting cytokinesis, which is cell division. These hormones are essential for creating new cells and tissues throughout the plant. One of their most valuable properties is their ability to delay leaf senescence, or aging, keeping leaves green and functional for longer periods. Cytokinins also promote the growth of lateral buds, encouraging branching in plants. These hormones work in close partnership with auxins to achieve balanced growth. While auxins are produced mainly in shoots, cytokinins are synthesized primarily in the roots. The ratio between these two hormones helps control the shoot-to-root ratio, determining whether a plant invests more energy in above-ground growth or root development. Abscisic acid, commonly abbreviated as ABA, is often called the stress hormone because it helps plants survive difficult environmental conditions. Its most critical function is triggering stomatal closure during drought conditions, which prevents excessive water loss through the leaf pores. ABA also inhibits seed germination, ensuring that seeds don't sprout during unfavorable conditions. It promotes dormancy in both seeds and buds, allowing plants to wait out harsh periods like winter. Unlike the growth-promoting hormones we discussed earlier, ABA generally acts as a growth inhibitor, antagonizing the effects of hormones like gibberellins. It also triggers leaf abscission, or leaf dropping, during stress, helping the plant conserve water and resources. Ethylene is unique among plant hormones because it's the only one that exists as a gas at room temperature. Its most well-known function is promoting fruit ripening, which is why placing a ripe banana next to green fruit can speed up ripening. The ethylene gas released by the ripe fruit triggers ripening in nearby fruits. This hormone also causes senescence in flowers, leading to petal wilting and drop. Ethylene promotes both leaf and fruit abscission, the process where these organs separate from the plant. Plants produce ethylene in response to mechanical stress, such as wind or physical damage. In some plant species, ethylene can also stimulate seed germination, adding to its diverse range of effects. plant hormones rarely work in isolation. Instead, they interact with each other to fine-tune plant responses. The balance between auxins and cytokinins is crucial for determining whether a plant develops more roots or more shoots. In tissue culture, scientists manipulate this ratio to control organ formation and produce specific plant structures. Gibberellins and abscisic acid work in opposition to each other. Gibberellins promote seed germination and growth, while a BA maintains dormancy and inhibits these processes. This opposition allows plants to germinate only when conditions are favorable. There's also a connection between auxins and ethylene. When auxin levels become very high, they can trigger the production of ethylene, which then leads to effects like fruit ripening and organ abscission. Understanding plant hormones has led to numerous practical applications in agriculture. Gibberellins are used extensively in the grape and citrus industries to increase stem length and promote the production of seedless fruits, making them more commercially valuable. Synthetic auxins have become important tools for weed control. These artificial hormones act as selective herbicides, causing uncontrolled, lethal growth in broadleaf weeds while leaving grasses relatively unharmed. In the fruit industry, ethylene inhibitors are applied to delay ripening during transport and storage, ensuring that produce reaches consumers in optimal condition. Finally, in plant propagation through tissue culture, scientists carefully control the ratio of auxins to cytokinins to direct the formation of specific organs, whether roots, shoots, or other structures, allowing for efficient multiplication of valuable plant varieties. Let's review the key concepts from this lesson. First, plant hormones are chemical messengers that regulate growth and development throughout the plant, even though they're present in very low concentrations. Second, there are 5 major classes of plant hormones, each with distinct roles. Auxins control cell elongation and tropisms, gibberellins promote stem growth and germination, cytokinins stimulate cell division, abscisic acid helps plants cope with stress, and ethylene triggers ripening and senescence. Third, these hormones don't work independently. Their interactions and the ratios between them determine specific plant responses and developmental outcomes. Finally, our understanding of how these hormones work has enabled numerous agricultural applications, from enhancing crop yields to controlling weeds and managing fruit storage. Plant hormones truly orchestrate the symphony of plant life, coordinating complex processes that allow plants to grow, adapt, and thrive in their environments.
If you like this video, hit that like button and don't forget to subscribe. Visit codelucky.com for more such useful content.